thank you all for coming to our talks today. I'm going to be talking about my work studying how proteins behave. So the reason why we care about proteins is that they're critical biomolecules that help the reactions in our bodies occur, which are going to keep us healthy and alive. Now, proteins' ability to perform those functions is dependent on their structure. And because proteins are very complicated compounds, their structure can change. They can unfold, which can cause aggregation and other types of outcomes. And sometimes this misfolding is associated with disease, like Alzheimer's disease. It's also known that when proteins come into contact with foreign surfaces, like medical implants, they can also undergo conformational changes. And while these changes might not cause Alzheimer's disease, they can cause other important outcomes. And so it's important for us to understand what makes them change their conformation. And as a physical chemist, that's exactly what I'm working on doing. Now, how can we go about measuring protein unfolding? Well, one thing we might want to do is be able to distinguish the folded and unfolded states of a protein. One way we can do that is we can label our protein with two fluorescent dyes. This will allow us to see them on a microscope. And if we can measure the distance between these two dyes, we can distinguish these two conformations of the protein. However, this method of solving this problem is a bit complicated due to the fact that light microscopy is limited to a resolution of about 200 nanometers. Now, while that is much smaller than most cells in our bodies, proteins are really tiny, and they're even smaller. And so if you're trying to distinguish two emitters that are on that tiny protein, it becomes difficult with this resolution limit. What this looks like? Well, if you have a single dye, you look at it on a microscope, you end up seeing an enlarged emission pattern. Now, if you are truly looking at a single emitter, that emission pattern is essentially a mountain. And we can fit that mountain with a mathematical function, find the peak of it, and that can give us a localization precision of much less than the diffraction limit of light, of around below 20 nanometers. However, when you have two emitters that are in this same resolution limited area, their mountain structure starts to overlap, and it becomes difficult to distinguish one peak from the other, which is of course going to make measuring our protein unfolding difficult. Now, this problem was a really important scientific problem. In 2014, the Nobel Prize in Chemistry was awarded to scientists who were able to overcome this problem and find a way to distinguish these two peaks. I'm going to talk about one way that you can do this. And the method that I've been using takes advantage of the natural process of photobleaching. If you've ever left fabric out in the sun for too long, you might notice that its color tends to fade over the course of several months or years. And the reason for that is at the single dye level, those dyes are each turning off as they become chemically damaged and losing their ability to give off light and color. What this looks like at the single molecule level is decreases in intensity over time. So I have multi-labeled proteins here with multiple dyes on them, and if we watch them over time, we see as they photobleach, they disappear. If we look at the intensity trace of one of these protein molecules over time, you can actually identify these distinct states when we have two dye on, one dye on, and then nothing left at all. We can extract images from these movies, and if we relate that back to our mountain analogy, by doing image subtraction, we can subtract the two mountain image minus the one mountain image to isolate the first mountain that was off. We're able to localize the dyes in the single mountain images, and that allows us to get a distance measurement between the two. Now, to give you an example about how we've applied this methodology to a real system, I looked at proteins in both crowded and uncrowded conditions on surfaces. What you might expect in a crowded surface is that a protein might be very surrounded by other things and unable to move around, just like if you're in a crowded room. But when you're not in a crowded situation, you have the freedom to unfold and stretch, as illustrated here. So in an uncrowded condition, you would expect to see larger interdye distances, and in a crowded condition, smaller interdye distances. When we applied this technique, that's exactly what we saw. And so in conclusion here, we're able to resolve single protein unfolding despite the resolution limit of light microscopy, and that allows us to distinguish two populations of proteins. Now, how does this relate to the big picture? Well, my work specifically, I'm currently working on applying this technique to unfolding on relevant biomaterial surfaces. And the hope is that in the future, this can inform the intelligent design of biomedical materials that will be used in our bodies in the future. Thank you all for listening.